So instead of talking, if I can get real dark, I'll show you a movie. Real dark, full dark, as before. Please. Yeah, you can see. So the movie that I'm showing you is a clip that we made 12 years ago of the structure and the function of bacterial ribosomes. It's based fully on crystallographic results obtained by us and by other groups with interpolation between them. It's available on YouTube for those of you that want to see it. Also, it's important to say that it's available on YouTube for the last 10 years, I think, or something like this. I didn't put it there, but somebody did. Because during all this time, nobody complained. Nobody wrote to me, it's wrong, so it must be right, isn't it? Scientists won't let it just stand. Anyway, we did it together with two students in the Art Academy, and the animators, and they decided that the cell has to be green, and the messenger has to come to the small subunit. This is their imagination. But otherwise, everything is based fully on structures. Okay, so here their DNA is coming to the small subunit that look, takes it. Look. Now, did you see it took it? This little motion that you saw is very little in the movie, but energetically it's very costly. So here is now the messenger around the narrow part of the small subunit, and this blue thing here is initiation factor. Initiation factor is coming from the cell. It's not part of the ribosome. In bacteria, they are free. In human, it's a very complex machinery. The duty of this factor is to tell the ribosome start. Otherwise, it doesn't know to start, so it initiates. So here is initiation factor number 3, and it makes sure that the messenger is bound correctly, and the first tRNA is associated correctly. It's brought with initiation factor 2. Now the factors are happy, they go away, and the large subunit can come and bind. You see the tRNA is now in its position, the amino acid is here, and the large subunit comes and associates with the small one by a surface complementarity, but also makes chemical bridges by conformational changes. Here are bridges that are made, 13 of them. So now we have an active ribosome, and we can look at it from solvent side. Here is the large subunit above the small one. tRNAs are being brought by elongation factors, and they are being decoded. Peptide bond is being made, and they are taken out, and the ribosome helps. In, out. You will see in a minute, I'll tell you 40 times a second. Look how fast. Now we took away the large subunit so that you can see that there is a bond being made and that the newly born protein goes into a tunnel. Now we are back to see the whole ribosome. You see more and more tRNAs are brought by elongation factors. This is the name of those blue ones. The protein is now starting to be large enough to come out, and the process continues until there is a stop codon. Says now the protein is finished. Everything is complete. It's okay. And then this yellow thing, which are other factors like recycling and release factors, they come in, replace the tRNA. The two subunits can dissociate. The protein can come out and go to where it needs, you see? And the tRNAs can look for the next job. This is the whole story. This is what we did for 20 years until we found this out. I just want to tell you that I think especially interesting for the biomedical and chemists, that it's not, the ribosome is not something undefined. It has, in bacteria, 250,000, 250,000 atoms, and we know the position of each of them. So what you see here rotating is the small subunit that is decoding. It's made of ribosomal RNA, which is 60% of its mass, and many proteins, 20 to 21 proteins, depending on the bacteria. Each of them is shown in a different color. Molecular weight, for those that are interested, is written here, 0.85 megadaltons, 1,600 nucleotides. It sediments as 30S, and this is its nickname. So what's important is that we know each position of each atom. That's really very important for the continuation. And that reminding you that this small subunit does the decoding. The large subunit makes the peptide bond, the next, and so on. It's a polysome. It also protects the newly born protein. Sediments is 50S, the color code is the same, and the size, of course, is larger, 1.5 megadaltons, 3,000 nucleotides, and up to 34 different proteins. So, to remind you what you saw in the movie with a little bit more detail, this is the small subunit in purple, and the large subunit in green and blue, and the active sites are the decoding and the peptide bond formation. So here, PTC is the peptide transferase center, peptide bond formation, and here is the decoding. The two tRNAs that are being decoded is P in the decoding, and A will come into it in a minute. Also, what you didn't see in the movie is that many ribosomes are reading the same messenger RNA, you see. This is a very, very old, I'm not sure that it's a good idea. Can you please put only half of the lights? Can you please put half of the lights? It was done really long ago, in 1969 or 70, at Oak Ridge, and I heard from one of the people here that has been there. So very, very fantastic electron microscopy. Messenger is going, many ribosome is reading, and even then they could see that the decoding is happening on the other side of the protein coming out. You see decoding here, and the protein comes out. And for years it was not understood until we determined the structure. So I'll tell you quickly about ribosomes. They act continuously. They make proteins all the time. You know that inside us today is not what we were inside 2 weeks ago, or even a week ago. Proteins are being replaced every 4 or 5 days. And it's not what we will be inside next week or the week after. They're working all the time. They can make up to 40 bonds in 1 second. Ladies and gentlemen, I was a good student. I had to make one bond in second year chemistry. This was my exercise. Took me 6 hours. And I was a good and fast student. I think that now peptide bonds can be made by 2 hours because chemistry developed a lot, but not 40 in a second. And not with such a mistake rate, 1 to 1 million. So maybe ribosome makes more mistakes, but this is what we can catch. Ribosomes are universal. And that's really important. They function in a very similar, almost identical way in all cells, in bacteria and in elephants, in the brain and in the stomach. They do, what do I call function? And now comes the question. They decode the genetic code, and they make the protein in the same way. Maybe exactly the same way, but if not, at least almost the same way. Maybe they do other things. There are some other ideas, but this is these are the main functions of the ribosome. So there are also many of them. They work, as I said, all the time. In the liver, for instance, there could be 5 or 6 millions in one cell. And in bacteria, about 80 to 100,000. So as I told you, I'm a chemist. I am a scientist. And I did not expect that any translational outcome will be generated by our studies. But my bonus is that something like this happened. So you can imagine that a ribosome is so important. Stopping the ribosome is a good way to stop the life. If we talk about pathogens, it means bacteria that cause diseases. Stopping their life is by stopping the ribosome. And it was known when I started, but even I didn't expect that I'll understand that or see that. My bonus, we know how all the antibiotics that paralyze ribosomes are working.